Invitarnos. Entiendo que esto es un panel de discusión sobre la película y nos daría gusto discutir la película. No sé si ya la vieron o la van a ver. No sé cómo lo tengan organizado para su congreso.
Yes, perfect. So we have some minutes to interact with you, and there are some questions that we are.
Sí, tenemos algunos minutos para poder interactuar con ustedes y hay algunas preguntas que estamos recibiendo en este periodo. Desde que la Sociedad Mexicana de Oncología lanzó esta comunicación mejorada para reunir a los oncólogos. Tenemos algunas preguntas. Y por favor, doctora Cabrera, doctor Bargallón, doctor Allison, doctor Sharma, nos sentimos muy honrados de tenerlos aquí en nuestro Congreso en México, hablando de cosas tan importantes. Me gustaría ahora hablar de algo intangible. ¿Cuándo empezó esto? ¿Empezó con su matrimonio o con alguna idea de inmunología y oncología? ¿Esto empezó con el amor? Pues sí, empezó hace mucho tiempo. Yo había estado trabajando en el bloqueo de punto de control desde hacía cierto tiempo para ciencia básica. Estaba tratando de llevarlo a la clínica y la doctora Sharma era clínica y estaba involucrada en el diseño de los estudios y en trabajar con compañías para realizar el trabajo inicial. Entonces, pues nos conocimos. Supongo que así fue, ¿no? Sí, saben, esto comenzó como una gran colaboración científica, tratándonos de asegurar que la ciencia que teníamos en el laboratorio, que era curar ratones, se pudiera trasladar a curar gente. Así que fue así como empezamos a hacer los estudios, a probar los agentes en la clínica, y fue el primer estudio clínico que comenzó mostrando la eficacia de la terapia de punto de control inmunitario. O sea que empezó todo primero como una colaboración científica, después empezamos a trabajar juntos, ¿no? Como que esas cosas las hicimos separados, después se unieron y empezamos a trabajar juntos y pues una cosa llevó a la otra. El resto es historia.
So I was asking you, after you win this award, what do you think? Maybe this investigation line is over? Maybe like game over? The prize have already...
No, tip of the iceberg.
Yeah. No, we're, the best...
Era como la punta del iceberg. La mejor respuesta es que el bloqueo de punto de control ahora es una combinación que se ha utilizado para melanoma, en donde la respuesta es como del 50% y los otros cánceres pues es más bajo, 20, 30%. En algunos cánceres, glioblastoma, cáncer pancreático, no hay respuesta para nada, entonces tenemos mucho trabajo por hacer. Pero la buena noticia es que creo que sabemos cómo se ve una buena respuesta. Y entonces sabemos, tenemos mucho más por aprender, pero sabemos cuáles pacientes con sus células T pueden estar beneficiados. Quizá es cuestión de ver a los pacientes en forma individual, incluso a los cánceres en particular, y pues lograr añadir lo que falte. Quizá si no cambiamos la firma mieloide, pues habrá que tratar de cambiarla, agregar más células asesinas, y si eso es el caso, pues agregar más células T. En fin, sabemos cómo debe de verse al final. Entonces es cuestión ahora de empezar a unir los componentes. Sí, creo que nos falta mucho trabajo por hacer. Claro que no estamos curando al 100% de los pacientes. Esa debiera ser nuestra meta. Exactamente. Entonces nos queda mucho por hacer en todos los tipos tumorales. Hemos hecho grandes avances en algunos tipos tumorales, pero en otros no funciona tan bien. Entonces estamos haciendo mucha investigación translacional con estudios clínicos y lograr una comprensión mecanística de cómo combinar diferentes modalidades terapéuticas. Muchísimas gracias por su tiempo. Ha sido un honor tenerlos con nosotros y quisiera que nos respondan desde el punto de vista de la vida y también desde el punto de vista científico. ¿Qué pasa con las personas que encuentran barreras en que no hay financiamiento, rechazo? Y luego, en segundo lugar, ¿qué pasa con el cáncer de mama? Porque es una enorme cantidad de esfuerzo, el esfuerzo de riñón, el refuerzo de, el cáncer de riñón, el cáncer melanoma. Bueno, primero queremos alentar mucho a los jóvenes allá afuera. Sabemos que están trabajando arduamente, sabemos que el financiamiento no siempre es fácil y tampoco lo es el trabajo. Nos adentramos en la ciencia y en la medicina porque es una pasión, es algo que nos impulsa, algo que nos encanta hacer. Tenemos que estar preparados para los altibajos en la ciencia. No todo trabajo experimental sale exitoso, no todo proyecto se financia y no tampoco se logra el éxito, pero la cuestión es hacer algo que a uno nos encanta. Mientras que tengas la pasión y que tengas la energía para seguir persiguiendo tus sueños, hay que seguirlo haciendo y esperemos tener excelentes maestros que nos sirvan como tutores. Muchas veces tus mentores te pueden elevar el espíritu, te dan orientación, guía y hay que seguir haciendo lo que uno adora. Si la ciencia y la medicina es algo que te gusta hacer, es una especie de vocación, es un llamado, así es como me lo explica mi mentor, es un llamado a hacer lo que siempre queremos hacer. Y en términos del cáncer de mama, tengo que decirles que me da mucho gusto ver que hay una aprobación de la FDA en cáncer de mama para PDL negativo, triple positivo, en la combinación mencionada, y ahora solo tenemos que lograr la aprobación para el grupo PDL negativo. Y esto también es importante sobre cómo pensemos en otros tumores, porque hay que convertir los denominados tumores fríos, que tienen pocas células inmunes y que son PDL negativos, a volverse tumores calientes con más células T infiltrantes y que se vuelvan, esperemos, positivos. Esto sigue siendo trabajo que estamos haciendo no solo en el de mama, sino también en otros tipos de tumor.
You're free to do it.
Well, I think that all your investigation is really impressive.
Creo que toda su investigación es muy impresionante. Nos sentimos muy honrados de tenerlos aquí, a los dos, juntos. Nos encantaría estar allá con ustedes. Sí, claro, están aquí. Pero lo que realmente quiero saber es, sobre todo en pediatría, en leucemia, la inmunoterapia para leucemia, hay mucha investigación que se está llevando a cabo. Esa es mi meta realmente. Es excelente, creo que eso es una meta excelente y muchas de las células CAR-T ofrecen gran promesa en malignidades, en leucemia, así que esperemos poder llegar a la terapia de control inmunitario y claro, hay que hacer mucho más trabajo en cáncer pediátrico. Con todo mi corazón, yo respaldo el trabajo que ustedes hacen.
Sabemos que cuando hay tratamientos nuevos o hay un giro muy importante en terapéuticas, sobre todo a nivel de oncología, existe cierta resistencia en tomar estas nuevas pautas. A lo largo de toda su investigación, ¿cuál ha sido la mayor barrera, independientemente de la económica, en cuanto a proceder con la investigación en inmunooncología?
Well, the first was just getting to do it at all.
Bueno, lo primero fue pues hacerlo, porque la gente era tan escéptica que me llevó 4 o 5 años de acudir a compañía tras compañía tras compañía para que la gente la convenciera yo de probarlo siquiera una vez, ¿no? Y todavía no probarlo todo, ¿no? Eso fue lo difícil, pero yo creo que los obstáculos ahora son un poco más sutiles. Tenemos que obtener que las personas hagan más ciencia y que analicen biopsias y que permitan que cambie todo y no nada más buscar una señal clínica. Y claro, esto es una pena porque hay muchos pacientes que se están desperdiciando desde el punto de vista que no se les trata con la combinación que funciona y entonces podríamos tener una señal que por lo menos nos indicara qué conviene utilizar como tratamiento la próxima vez y que sí sirviera. Es bastante frustrante. Yo creo que los principales obstáculos con los que lidiamos ahora son estrategias y combinaciones racionales. El primer obstáculo al que se enfrentó Jim fue que empezara la inmunooncología para que se viera que podía hacer una diferencia en la vida de las pacientes y que fuera aprobada por la FDA. Esto ocurrió en el 2011, ya lleva 9 años y ahora todo tiene que ver con que tengamos las combinaciones y las estrategias racionales y que podamos obtener datos de los humanos, que sepamos qué pasa con las respuestas inmunitarias humanas para continuar con los siguientes blancos. Así que esto es lo que perseguimos en forma de campo. A nivel de individual, digamos, el premio Nobel ciertamente nos ha abierto muchas puertas y avenidas para que podamos tener una plataforma en la que podamos ponernos de pie y hacer avanzar esta área gracias a esto. Pero antes de que eso ocurriera a nivel individual, se trataba de convencer a muchos otros clínicos e investigadores de que esto era pues una trayectoria valiosa que había que seguir. Entonces fueron esos primeros estudios clínicos los que resultaron muy difíciles, sobre todo por la acumulación de pacientes.
La inmunooncología debe de empezarse a enseñar ahora a las nuevas generaciones, tan temprano como desde la medicina, o tan temprano como desde high school.
As early as learning
Tan
chemotherapy
temprano en nuestro...
immunology should be taught. Immunology should be taught when you learn
La inmunología se debería de enseñar, al igual que la biología, ¿no? Como el sistema sanguíneo, como cualquier otro sistema orgánico. Yo creo que en las clases de pregrado se dan cursos básicos de inmunología y cuando llegas a la escuela de medicina, claro que debería de haber una clase de oncoinmunología, al igual que se enseñan muchos otros cursos. Mientras más pronto mejor, porque entonces creo que se tendría más médicos experimentados que pudieran dar terapia a los pacientes.
Bit complicated being a low middle income country and look out all the trials and the combinations and the magnificent results and get to the clinics and guess that your patients can't not afford those drugs. So what are you thinking about from the leadership of prime?
¿Qué piensan ustedes del liderazgo a partir del premio Nobel? ¿Creen que esto abra el acceso al resto del mundo? Bueno, nosotros fungimos en muchos comités y tenemos conversaciones con diferentes aliados del sector farma y la accesibilidad a los medicamentos es un gran problema. No queremos tener que ir a un centro importantísimo de los Estados Unidos, un centro médico para poder tener acceso a los medicamentos. Digo, cada país tiene que tener su propio sistema de seguros médicos y tiene que tener una forma en que se pueda proporcionar los medicamentos en cuanto a cómo se paga a cada compañía. Cada país tiene problemas diferentes. Yo sé que algunos países están tratando de producir los fármacos a través de lo que llaman ellos los biosimilares. O sea, no es lo mismo, por ejemplo, que utilizar el fármaco de Merck o de Genentech o de Bristol Myers. sino que es un similar que puede ayudar al bloqueo de PD1, PDL1. Están tratando de producir los medicamentos en sus países y esto puede influir en términos del acceso. Yo creo que estas cosas se tienen que abordar no solo desde el punto de vista científico, sino que también las dependencias gubernamentales se tienen que involucrar, también las agencias gubernamentales. O sea, yo me gustaría usar la palabra democratizar, ¿no? Para que mucha gente la pueda obtener, la más posible en todo el mundo. Sí, la atención para el cáncer debería de ser equitativa. Todo el mundo debería tener acceso a un cuidado adecuado para el cáncer. Sí, claro. Yo quisiera saber su opinión sobre la interacción entre la inmunoterapia y la radioterapia. Pues hay mucho trabajo. Aquí lo que hay que saber es que hay que cambiar la forma en que se aplica la radioterapia. Si se hace de manera fraccionada, en donde se mata a las células y el sistema inmunitario entra para lidiar con la inflamación y las células T empiezan a tratarse de recuperar y luego se irradia otra vez y se matan todas las células, eso no funciona tan bien. Hay varias metas, o sea, la meta de la radiación ha sido mata a todas las células. Yo he visto cómo funciona, ¿no? Pero ahora es más bien mata apenas a las suficientes para que él termine el trabajo, el sistema inmunitario y no acabes con el sistema inmunitario en el trayecto. O sea, hay mucho trabajo por hacer, pero creo que hay esperanza. Sí, yo creo que la radioterapia se puede usar para mejorar y reforzar la respuesta tumoral y mejorar la presentación de antígeno con células dendríticas y macrófagos, pero no se puede administrar en una forma tan fuerte o tan frecuente que se mate a las células presentadoras de antígenos o células dendríticas. Se trata de encontrar nuevas maneras de administrar la radioterapia que pueda lograr sinergia con los agentes inmunoterapéuticos.
¿En algún futuro la inmunoterapia sustituya a la quimioterapia?
No, pensamos que de hecho que la inmunoterapia se podría combinar con la quimioterapia de una forma en que no se tenga que administrar tanta quimioterapia, lo cual sería bueno porque entonces no se tendría la toxicidad de la quimioterapia y también en algunos ámbitos podríamos administrar quimioterapia al principio, en las primeras etapas de la enfermedad y si fuera necesario también administrar quimioterapia en ese momento, pero la idea es mantener la memoria de respuesta para evitar recidiva de la enfermedad o recaídas, es decir, que sería o inmunoterapia e inmunoterapia más quimioterapia, pero cada tipo de enfermedad, cada etapa de la enfermedad puede requerir de un tratamiento diferente. Yo creo que la quimioterapia como tratamiento por sí mismo va a desaparecer, pero creo que cuando lo pensamos por primera vez, todo el proceso comenzaba preparando a las células T por la muerte tumoral, por la inflamación y si se tiene un tumor que uno sabe que responde a algún agente quimioterapéutico, pues a lo mejor se podría bajar la dosis para que sea menos tóxica y aquí otra vez es la idea que hay que matar suficientes células para que entre el sistema inmunitario. Esa es otra manera de cebar, de preparar más eficientemente. Si se dan las quimioterapias por sí solas, pues se está dependiendo, o sea, a que las células tumorales se mueran, por decirlo así, en el pasillo. Hay que saber qué es lo que está ocurriendo. Esto es muy importante con la radiación y la quimioterapia. Y eso es como la dosis de cebado, ¿no? Es la dosis suficiente y hay que desarrollar en base a eso. Yo no creo que vaya a desaparecer así, pero va a cambiar fundamentalmente. O sea, no se pueden matar todas las células tumorales, no se puede dar tanta toxicidad, solo matar suficientes para que se provoque inflamación y entre el sistema inmunitario. En algunas enfermedades la quimioterapia funciona muy bien. Si piensas en los tumores de célula germinal, por ejemplo, uno no quería eliminar la quimioterapia si funciona tan bien. Otra pregunta. Su enfoque es más bien en oncología, pero ¿tienen alguna investigación u otro paso, otro tipo de tratamiento que pueda contribuir ¿O creen que se pudiera ganar otro premio Nobel con algo más? Quizá con terapias combinadas, ¿o qué piensan que serán los próximos pasos para el futuro cercano de la oncología? Yo creo que el futuro cercano será ciertamente algo con inmunoterapia. La cosa es saber qué se va a combinar con la inmunoterapia. A lo mejor puede ser modificadores epigenéticos para que se pueda epigenéticamente no solo alterar las células tumorales, sino también las células inmunitarias epigenéticamente para tener mejores células T o células mieloides mejores o células dendríticas para que se pueda manejar la respuesta. Esto tendría que ver, por ejemplo, con la modificación epigenética, que creo que habría que considerar. Y ciertamente también dentro del espacio de la ingeniería se está haciendo ingeniería para crear mejores proteínas, ya no solamente células CAR-T, sino células específicas que puedan tener como blanco ya sea el antígeno tumoral y la célula T al mismo tiempo, para que esta célula T se lleve más cerca de donde se expresa el antígeno. Creo que estos son el tipo de cosas que vamos a ver que lleguen en el futuro. La combinación con inmunoterapia y me parece que dijo virus. ¿La combinación con qué? Perdón, ¿inmunoterapia y qué? Virus oncolíticos. Sí, absolutamente. Los estudios clínicos están analizando esto y van a ver qué virus oncolíticos, porque hay diferentes bases, ya sea adenovirus, virus de polio, etcétera. Son cosas que hay que combinar. Ya se probó el medicamento de Amgen y por supuesto que es posible tener tratamientos virales, no solamente como columna vertebral, sino que se pueden dar en forma intratumoral o intravenosa, y todos estos son estudios que están en curso en este momento, pero yo creo que los virus oncolíticos tienen una gran oportunidad de preparar a la respuesta inmune y también impulsarla contra los componentes virales, que también nos daría reactividad cruzada con la expresión antigénica. El abordaje del virus no solamente permite matar las células tumorales quizá de una manera mucho más limpia, sino que también se podrían empaquetar ciertos genes en el virus de tal forma que se pueda inducir moléculas neuroestimulatorias en el tejido y al ir muriendo que se aumentara su presencia en el tejido. O sea, la capacidad de hacer ingeniería a los virus oncolíticos es gigantesca y yo creo que esto va a ser muy, muy importante. En mi laboratorio estamos trabajando y hemos estado trabajando en esto un poco. Tengo una pregunta de Monterrey, México.
Buenas noches. En nuestra ciudad, Monterrey, iban a estar ustedes invitados al Congreso. Esperemos tenerlos muy pronto. Muchas felicidades. ¿Ustedes sienten que ya le entendieron a la inmunoterapia o están rascando la superficie apenas?
Well, we're beginning to understand it.
Estamos empezando a entenderla. No, estamos arañando la superficie.
Y la otra pregunta, en nuestros países donde el acceso es un problema, ¿sería relevante conducir estudios clínicos buscando dosis mucho menores de inmunoterapia combinadas con otras formas de tratamiento? ¿Y habría modo o ideas para conseguir el funding, que obviamente no vendría probablemente de la industria, sino de otros fondos independientes, para conducir investigación con dosis más accesibles de medicamento?
So I think you're absolutely right. One, we have to try.
Creo que tiene toda la razón. Hay que manejar diferentes esquemas y diferentes dosis por la toxicidad. Si usted se fija, algunas de las aprobaciones, por ejemplo, con ipilimumab está aprobado a 1 miligramo por kilogramo, otros se aprueban a 3 miligramos por kilogramo, pero esto es algo que hay que evaluar dependiendo del tipo de tumor y quizá la población de pacientes, o quizás si se va a combinar con algo más. Y también la accesibilidad en países que quizá no tengan las alianzas con el mundo de farma para hacer los estudios clínicos. Esto es algo que tenemos que ir relacionando. En los simposios de oncología tenemos, por ejemplo, el de la ACR, el de la ASCO, el de la Sociedad de Inmunoterapia y Cáncer. Todos estos son grupos internacionales que van a unirse a discutir esto como un organismo, como un órgano para ver cómo abrir los estudios clínicos en otros sitios, aunque se han abierto en Estados Unidos. Pues, por ejemplo, los mismos órganos pueden financiar, entonces habría que encontrar la forma en que se pueda movilizar. Claro que con el COVID todos los estudios clínicos tuvieron que cerrarse, incluso los de Estados Unidos, o sea que estamos apenas empezando a ponerlos en marcha nuevamente. Muchas gracias por esta parte final de esta reunión. En verdad la apreciamos y estoy seguro de que nuestros socios
the immunotherapy for. At the final, I ask to you, both of you, that a final message for our people in Mexico, our oncologists.
¿Cuál es el mensaje final para los oncólogos en México que siguen luchando con el melanoma o el cáncer? Quizá puedan recibir sus palabras, quizá los comparen a ustedes con superhéroes. Quisiéramos que todos sintieran el éxito de este trabajo, porque lo tenemos que hacer entre todos nosotros. No somos solo nosotros dos, hemos trabajado con equipos grandes por todo el mundo. Así que esperamos que ustedes disfruten mucho su sesión y que generen nuevos tratamientos que en un momento dado nos puedan enseñar a nosotros. Sí, muchas gracias por esta oportunidad de conocerlos y esperemos ahora tener la oportunidad algún día de conocerlos personalmente. Muchas gracias. Reciban nuestra admiración, nuestra gratitud y un gran abrazo de México a Houston. Hasta luego. Muchas gracias a todos.
Ahora podemos pasar a que ustedes conozcan la historia, y realmente es un cortometraje sumamente emotivo, sumamente interesante para quienes somos médicos, quienes somos oncólogos. Lo van a vivir de una manera sumamente intensa. Los dejamos con ellas y al final regresamos con ustedes. Muchas gracias.
It's a part of Texas that was still majority Hispanic. Football reigned, everybody, Friday nights, that was what it was all about. And it was an oil boom town, too, although there was a lot of agriculture as well. My mother's family's been there since probably the 1840s, I think. My dad was a doctor who, took care of people in the neighborhood, did house calls. My father moved to Dallas in the 30s sometime. I don't remember exactly when, but I was born in 1948, and we lived, I think, here, actually, because there was a washing machine out in the yard, and I got my arm caught in the ringer one time. It pulled me up and, got stuck, and I was hanging, by my arm, and some people, neighbors came out. I have two brothers, Murphy and Mike.
My mother, she was a housewife. She was a good influence on us. Just kind of give us a little discipline. We get to wash the dishes on Sunday and things like that. Of course, that may have been a little bit before Jim's time. I think he got out of that.
didn't really know what was going on in those days. Nobody talked about it. All I knew, she was sick, and she had these burns on her throat, and you know, she was in bed a lot. I remember one day I had some good friends, had a kid my age, and his mother was going to take us to the swimming pool. I'm just about to get in the car, and somebody came running out and said, "Hey, you got to come back. You can't go," you know. So I went in, and she was dying, and so that made her. So she died that morning. And I found out later, I really didn't know what it was, but I found out later that it was lymphoma. She'd been getting radiation therapy, hence the burns on her neck. I sat on the bed, you know, with her and held her hand while she died. You know, there was certainly a big hole, a lot of loneliness. I was 11 years old when my mom died. My father was just heartbroken. You know, he seemed to kind of lose interest in things. Anyway, he was gone a lot. I ended up living with sort of a foster home. I mean, not in the sense that I was giving away everything, but a counselor from high school teamed me up with a family who had a boy about my age, and so I spent when my dad was away. I stayed with them all the time. I like to wander around in the woods. I lived right at the edge of town. I used to wander in the woods by my home. And I just found it was comforting to have a harmonica and just toot away, you know.
On behalf of the Nobel Assembly at Karolinska Institute, I wish to convey to you our warmest congratulations. May I now ask you to step forward to receive the Nobel Prize from the hands of His Majesty the King.
Innovation is extremely difficult.
We were all very skeptical.
Many people said, "Jim Allison, you're wasting your time."
I knew that this is a way to cure cancer. I knew that that was a way. And I'll be damned if I'm going to let that just lay around. Well, I guess my dad encouraged me to mess around with my Gilbert chemistry set in the garage, you know, and dissect frogs and horned toads and things.
He told me about the bombs he used to make. So he would construct these little bombs out in the woods, and he said, you know, in this very Jim way, he's, you know, how, you know, when you talk with scientists, you know, later in life, you realize a lot of us were bomb makers in the beginning.
When I started learning about what science really was and how to do it, you know, I started doing more intelligent things than making bombs. One of my teachers who taught advanced mathematics, physics, chemistry, I thought he was one of the greatest teachers I ever had. But I tried to have a few conversations with him about evolution, selection of the fittest, Darwin. And he just said, no, you know. It's not real. I do not allow. We will not teach that here. We will not discuss it. I refused to take the course because I knew that there was no biology in it. The syllabus, there was no biology in it. So it was a pretty ugly time, and it was basically me versus him. I was still taking advanced physics from him while I was having this fight with the school, because he was the head of the science program. He was the one that made sure that evolution wasn't taught, and so he really did start taking it out on me. Back in those days, you could use boards to paddle students, so I seemed to be getting in trouble all the time and getting a couple of licks just for walking by, They just thought I was a troublemaker. I wasn't. I was just trying to do what was right. That's the way it is, If you don't agree with something, you need to stand your ground. And if it hurts, it hurts, That's the way it is,
When he was little, we said he was hard-headed. We never did call Jim this to his face, but, this guy, we called him Diamond Head, because his head was the hardest substance known to man.
Jim graduates from high school at age 16. He goes to the University of Texas in Austin, determined to become a biologist.
There was this knock on the door. Door opens and this scrawny kid pokes his head in and says, "You got a job?" I said, "There's one job left, but you may not like it. It's just a dishwasher." And he said, "I'll take it." We would take a piece of cheesecloth and put a little bit of acetone on it and rub off the number of each test tube. And literally there were thousands of them. And so he got the idea, well, maybe I could take those test tubes and spray them with a solid stream of acetone. This was back in the 60s, and just about everybody smoked then, including in the lab. Jim sprayed his test tubes with acetone and then sat back to have a cigarette. And boom! And we all run in from the lab, and here's Jim with a blackened face, and his eyebrows were all singed. I was about to fire him, but in the short time he'd been there, Jim had just got along and knew all the graduate students and the others, and they besieged me on his behalf. "Look, can you find something else for Jim to do? He's quite a character. Let's keep him around."
Well, Candy, won't you be my queen?
In the summer of 1966, he came for a fraternity party that I went to with someone else.
Together we can make honey, baby, that the world has never seen.
He was the most amazing thing I'd ever seen in my life. I never dated anyone else in college. We were together from then on. It was a really kind of magical time to be in college. It was so much fun, so much going on.
We were lucky enough to be in college in Austin just as everything was beginning to happen there. That really made it when Willie Nelson moved there. I think the first time I saw him live was playing on a flatbed truck at a Ford dealership on South Lamar in Austin.
But everyone was also real serious about their studies. Jim was passionate about science.
When I was at the University of Texas, the number one thing I learned in the lab was just how important it is just to persevere. Not be so disappointed you give up, just keep working.
We could never go anywhere without going by the lab, checking on this or that, experimenting process.
T cells were discovered when I was in college, and I took an undergraduate class, and my professor, Bill Mandy, taught one lecture about T cells because almost nothing was known about them.
The human immune system circulates in our blood and protects us from disease. We now know that at the center of the immune system is the T cell.
T cells are a type of white blood cell. And these are the cells that actually respond against viral infections, bacterial infections, and they start the fight to keep you healthy.
These cells that go all over your body and manage to protect you and not kill you. It's still, to me, just wondrous. I wanted to try to do something that Barry Kiddo always taught was, you should try to do things. If you're going to do this stuff, you ought to at least do things that help people. My mother had 3 brothers. One died from melanoma. In those days, melanoma was a death sentence, so he didn't even get therapy. When he was diagnosed, he just said, that's it. Had a really good time for the months that he had. The other brother was a former rancher who could roll a Bull Durham cigarette with one hand while he was on a horse, but he paid for that. He had lung cancer and died from that. I saw him in an oxygen tent when he got to the point that his emphysema or whatever was so bad, he needed help to breathe. Those made kind of an impression.
My own lab was not involved with cancer when Jim came to work with us. But he had had a tremendously difficult relationship with cancer. So at graduate school, he convinced me that perhaps we could look at something in the cancer field.
There was an enzyme called asparaginase that when injected into kids with leukemia, could induce remissions. I first thought they were cured, but it turned out they weren't. I mean, they were for a time, but then they always came back. My thesis project was to find other asparaginases from other bacteria that would be so different that they could be used in succession.
We pulled water out of the local stream and surveyed that for asparaginase activity. Jim purified that. And in mice, that worked very well indeed.
Jim's work on asparaginase convinces him the immune system plays an important role in responding to cancer.
I was a bigger kid growing up. I really ate way too much sugar. I know that now. I played a lot of sports. Basketball was my thing. I played my first year of high school and then injured my knee pretty darn bad. Kind of lucky that I did, because I couldn't play basketball anymore, but I joined the marching band, where I ended up meeting my soon-to-be husband. I was finishing up my graduate year. I had what, feels like bronchitis. You were coughing, breathing was a little bit of an issue. Progressively over the next couple of months, I didn't get better. I was walking up a hill one day and I could not take a deep breath. My mother worked for a primary care doctor, and she's like, "Oh, sweetie, when you get home, come on, see him, and we'll get this sorted out." Little did I know that just a few weeks before that, I was going to develop a mass growing out of my chest. And I thought it was breast cancer because of where it's located. So going to see her doctor, they noticed the tumor and they did a biopsy, and my world changed.
I went to Scripps because I wanted to learn about the immune system. I wanted to ask questions about how the immune system worked.
Strangely enough, there was a group of expatriate Texans there, and we would all go to a little bar called the Stingery. And there was a band there that he would sometimes play harmonica with.
Columbia Records was having their annual convention in San Diego, and Willie Nelson's record, The Red-Headed Stranger, had just gone platinum, so they decided to have a big party for him. The guy that was in the sheriff's department, was a real devotee of our band, came and told me about this, and said, "We're doing security for it." He let me go in, let me through security to go to this private party. Willie Nelson came over and he said, "Well, you know someplace we could go picking tomorrow night? Because I got tomorrow night off, and I'd sure like to play some music." So I said, "Well, it's talent night at the Stingery."
So we loaded up in our little VW camper. We walked in and people just fell on the floor.
Willie basically asked, "You guys mind if I play?" He had his drummer and his bass player and started playing. They played for 3 hours at least. And, it was a little dive bar, and there's about 8 people at this place, There was one payphone on the wall, and so those 8 or so went up, called people, and pretty soon the place filled up, and people called other people, and it was full, and people were out on the street. It was amazing. I didn't have to buy a beer at that place for about 2 years after that. That old college roommate of mine, he heard that MD Anderson was opening a new laboratory near Austin. To tell you the truth, the main reason I went there is because I thought I would have freedom.
The first time I connected with Jim was, I think, in about 1980. I was a postdoc in New Mexico. I went to MD Anderson, and they asked me to give a seminar. And they said, "Well, you're being live streamed to Smithville, Texas." And so I gave my seminar, and they said, "Do we have any questions from Smithville?" And I'm thinking, where in the hell is Smithville? And then this voice comes over with this, nice Texas accent and asks me this incredible question.
With a given molecule on a pathway, for example, the MAP kinase pathway, which, is
He
involved in
got right to the heart of, the hardest question he could ask about the science I was doing at the time. I consider Jim my brother, scientifically as well as personally. I would consider him my best friend for the last 30-odd years.
I really wanted to understand the immune system and everything, so I really wanted to know how T cells worked. And at the time, all we really knew was that T cells went around the body and took care of stuff that didn't hurt you.
Scientists know that T cells distinguish between healthy cells and threatening ones by using a molecule on their surface called the T cell receptor. But they don't know how the receptor works.
We didn't know how those white blood cells, these T cells, would actually recognize the virus as being different and needing to be eliminated. And that was the holy grail in the late 70s and early 80s. you know, all the top labs in immunology. That was the quest.
I read the literature, and there was a lot of stuff that was contradictory, a lot of stuff that seemed like fantasy. I could not possibly imagine how some of the mechanisms that were proposed actually worked. I said, well, either I'm a total moron and can't follow this complicated stuff, or people are just, you know, it's all nonsense. You know, so finally I said, you know, forget it. I'm not going to pay any attention to the literature.
Part of Jim's success is the fact that he's an iconoclast. You know, he doesn't care. He doesn't care that he's not following convention. He's following his own thoughts, his own motivations. He's willing to do it in the face of his colleagues not necessarily believing him.
You got to notice that Irv Weissman from Stanford, who was a very noted immunologist, was giving a lecture. So I drove 3 hours from Smithville into Houston. I was driving back, replaying his talk in my head. I remember it just sort of clicked, and I said, I know how to find the T-cell receptor.
Great scientists are creative people. They're not just focused on their detailed work. They're thinking beyond. There's a creativity to this work, and frankly, if not for the creative scientists, we would make little progress.
I saw miles and miles of Texas, all the stars up in the sky. I saw miles and miles of Texas.
Drove back that night, just rolling in my head and everything, and you know, the next few days, just lined out a path to find the T-cell receptor.
Over the next few months, Jim uses the latest genetic engineering techniques to test his hypothesis.
Work hard all day, get an incubation period or something, go to the beer hall, you know, knock down some beers, go back and finish the experiment. You know, go to sleep, get up, start it all over again. You know?
I think the guy is just incredibly passionate. So he'll spend a night going out to a honky-tonk, and he'll be out all night, and he'll drink, and he'll have a great time. And then there's that same energy that would put him in the lab at Smithville. They're doing a 36-hour experiment, and he's staying up for that. And he's just as driven and turned on by that whole experience of being in the lab.
We went to a conference in California. This is where you present your data. You're not well known enough to be invited to get up on the podium and give a talk at that point. You know, you're relegated to putting your work on a poster board. So Jim had a poster there, and I remember walking over to look at his poster, and we'd just kind of met recently then, and he pointed to this spot on the poster and says, "That's the T-cell receptor." And he was right.
His contributions to our understanding of the T-cell receptor, the molecule most centrally identified as the thing that recognizes the target and allows for it to be eliminated. Extremely important.
He was the first one to actually purify the molecule by which T cells recognize everything. This is how T cells recognize infectious disease. This is how T cells are recognizing things they shouldn't recognize when you get autoimmune diseases. So this was the seminal discovery.
Jim was invited to major immunology conferences, and it was recognized that this constituted a real breakthrough.
When they came back and said, Sharon, you have melanoma, I was completely shaken. Unbelievable. I couldn't believe that that was my life. I thought I was going to die.
Good job.
The doctor canceled all his patients and shut his office door and got on the phone and said, I have a 22-year-old patient here who needs to see you now. And he was on the phone all day, but I had an appointment with Sloan by the end of it. So then I saw Dr. Walchuk just a couple days later.
Sharon Belvin, when I first met her, was 23 years old. She was seen by an oncologist near her home in the New Jersey Shore, who called me and said, you need to see this young woman. So I see her, and it's a room full of very sad, family members trying to get their head around this seemingly unbelievable situation. We started her on chemotherapy very quickly thereafter.
So I had chemotherapy Monday, Tuesday, Wednesday, and got married that Saturday. We decided to go through the wedding because it's for better or worse. I mean, we were joking around a couple days before, and we were just like, okay, well, we are going to get the worst over with right in the beginning, and then the rest of life will be great. And Rob is really probably a saint, and don't tell him I said that. Who can look at somebody and say, here's a terminal diagnosis, and I'm going to marry you anyway?
In 1981, there was a bill that was introduced by a very conservative state senator from East Texas saying that creationism needed to be taught in schools.
One of his teachers from Alice had actually been elected as the state representative from Alice, not the science teacher, of course. And she called him up and said, someone has introduced a bill to outlaw the teaching of evolution in the public schools, and we've got a hearing coming up, and we need someone to come and talk.
She said, he's going to have 300 people here supporting the bill and opposing evolution. And so if we don't have somebody here speaking to the other side, it's likely to pass from committee out to the House floor, and God knows what's going to happen. I tried to find other people who would do it. I only found one other scientist that was willing to do that.
I'm here tonight speaking before this committee because I feel that in this highly technological age, an education in science is essential if members of our society are to be able to understand and deal with complex and diverse problems facing them. And I know from my
personal... Science is something that you can use. It's not just accounts for the past. It does that. It also allows you to understand what's going to happen in the future, to situations that you've never seen before, that have never arisen. That's what science is.
That is the chief problem with creationism, purporting to be a science. It cannot be used to construct explanations of newly observed fact. It does not give us any help whatsoever in understanding how bacteria acquire resistance, how tumor cells arise, or how they are eliminated by the immune system.
He talks about using the immune system to cure cancer at that point, even in 1981.
I said, I challenge this man to use his creation science, such as it is, to understand how a tumor escapes the immune system. It's hilarious, the limits of their arguments. Probably the best one was, if you put a Ford in a field, it rusts, don't turn it into no Cadillac. I said, oh, yeah, okay, I got it. Obviously, it can't be anything like evolution.
pretty soon after that, he got an offer from the University of California at Berkeley to go out there.
After the T-cell receptor, I was offered a full professorship at Berkeley, and I was really happy out at Smithfield, living in Austin, working in Smithfield, just doing science. It was as clean as it could get. I was doing exactly what I wanted to. I wasn't answering to anybody. I didn't have to do any administration or anything. And so the idea of going into some big-time place and having to, compete was a little scary. But an old friend of mine called me up and said, "What are you doing?" And I told him that I'm comfortable here. I'm happy here. He says, "Yeah, in 20 years, going to be sitting there with their feet up on the porch saying I could have been a contender." I kind of went, "Yeah, I understand what you mean." When I decide who to hire in my lab, I always am looking for one thing, really, and that's curiosity.
Berkeley drew me in a lot of different ways. It's filled with very smart people, but very freaky people, and they all sort of intermingle, and you don't ever know what's a crazy idea and what's a great idea. Even at Berkeley, Jim was a standout. He didn't look like the other people. Berkeley has its own conservative liberal side of it, which is that, you can be liberal up to a point, but Texan is actually too far. It's like you're carrying a gun and I don't know, all kinds of things that could be bad. And so Jim was like just over that line and represented just the, additional frontier even of Berkeley. But also immunology was not considered a science. The classic people said, "Oh, immunology, that's sort of a pseudo-discipline. That's really like almost like giving holistic medicine or something, right?" It's like, what is that? You don't even know what that is. So Jim's lab has a feel of being like a pirate ship. There are personalities doing their own crazy stuff, and yet there is a dedication to the purpose of driving the boat forward.
I was working a lot, not as hard as the people in the lab, of course. I mean, they were working all the time. But we also all partied together. I just had some incredible parties.
There is the rigid thought process that is like connect the dots, A to B to C to, like that. And that you do during the daytime. But I think there's a whole element of the idea of unwinding and not letting the mind go linear. And just saying, what if nothing we thought was right?
Jim's work on the T-cell receptor has sparked a race to discover more secrets of the T-cell. Soon a French scientist finds a new protein on the surface of T-cells called CTLA4. Most researchers assume that it activates the immune system like the T-cell receptor. But Jim doesn't assume anything.
The only right way to do science is not to do experiments that give you an answer that's consistent with what you think you ought to get, but do the killer experiment that can only be right if you're right.
Jim started looking at the data on CTLA4 critically, and he and Max Krummel, his graduate student, started saying, well, let's see what the real data is. If we do experiments, is that true or not?
So we just had this stupid question, like, what does it do? And it wasn't like just cure cancer. It's like, well, what's that do?
Are you shooting, Jim?
Yes.
Well, it's Christmas. It's 1991, and we're loading up the piñata here on Christmas Eve. Here's Robert.
Robert?
To study CTLA4 in a mouse, Krummel must build a mouse antibody, a Y-shaped protein that will trigger a reaction from CTLA4.
Yeah. Police car. Stand left, Robert's a police car.
Okay, let's go.
What made that whole 3-year stretch for me very difficult was that I didn't get any progress scientifically.
Robert's person, fourth birthday. Robert, look over here. Look, are you Batman?
And this is really common. For 3 years, nada. You know, like everything I did really didn't amount to anything.
Running laboratories, it's not easy to have people do the experiments you want them to do. I regret at times not being able to motivate people to do the kinds of experiments that I was sure needed to be done. But Jim has that ability to do that. So because of that, people in this lab have done things that were crazy at some times, but he also was able to get people to do the things that he was really passionate about.
While Max struggles to make his antibody, another lab beats him to it. Peter Lindsley, a scientist working with the drug company Bristol Myers Squibb, publishes results saying that CTLA-4 is an activating receptor. When it's engaged, it revs up the immune response against targets.
Then these papers came out and said it's a positive regulator. And so, you know, that attitude was, well, we've been scooped, you know, it's done. Except I said, I don't think so. There's something about, you know, I saw some holes in that paper, and so we're gonna test this fucker and see if it's really right.
You have this picture of Einstein sitting at the whiteboard, hair frayed, you know, writing equations that no one can see. Really what he's doing is saying there's a fundamental question, an assumption people have made that doesn't make sense if you look at this in a clear-eyed way. What if I step back here and just observe and say, these pieces of information need to be put together in a different way? I think that is what pioneers do, and weirdly, it's childlike.
This is a hard thing. It took me a while to be able to say this to people, but I think there was a formative moment when I was in Jim's office, and I actually started to cry. Because I'd been working on something for so long, and he told me something, again, a mistake that I'd made, and you know, I just started to cry. And then I got over that. I mean, there was a period of that you just have to get through and just say, okay, go back at it.
Crummell finally makes his CTLA-4 antibody and launches Jim's experiments. He quickly demonstrates that Lindsley's idea of how CTLA-4 works is wrong. The experiment proves that engaging CTLA-4 doesn't activate the immune response, it stops it.
Jim and Max Crummell, as well as Jeff Bluestone and his grad students, turned the thing completely around, said, nope, all these prior papers saying that CTLA-4 is another gas pedal, that's not correct. It's actually a brake.
Like Jim, I was able to figure out CTLA-4, and I was shocked. CTLA-4 was the wackiest thing in the world, right? Nobody believed that you would actually have a whole system in place to shut down the most important thing going on in your body, which is an immune response, and you're gonna shut this thing down and make sure it doesn't work.
The body does not want an over-exuberant immune system, because if we did have one, we would all be suffering from autoimmune disease.
Autoimmune diseases, like multiple sclerosis, are caused when the immune system attacks healthy cells. Jim's discovery prompts many immunologists to think about curing these diseases. They will engage CTLA-4 and stop T cells from going on the attack. Jim heads in a different direction.
There's this big part of the immune system that is built on stopping itself. Now, what the heck does that have to do with cancer?
Cancers We know they have mutations. We know they're different from normal. They should be visible to the immune system. And for a long time, we struggled. Why can't the immune system see cancer? It can see flu.
Jim theorizes that tumors have evolved to trick the immune system. They engage CTLA-4, turning on the T cells' brake. This stops T cells from attacking the tumors.
If you were a cancer, it would certainly be advantageous if you contacted that receptor and turned off all those T cells that might otherwise come to kill you. This would make sense. At the time, it was a crazy idea. Nobody thought tumors were trying to turn off the immune system. But he said, well, what if I block this? Why don't we see what that does to certain cancers?
Jim hypothesizes that if he inserts an antibody to block the connection between the tumor and CTLA-4, the T cell will be free to attack the tumor.
Jim had all these tumors in the freezer. So I was making antibodies as fast as I could, and a guy named Dana Leach came. He was a postdoc in lab, new postdoc, and Jim gave him the tumor models, and I gave him the antibody.
The student who was doing the experiment had put the tumors in the mice and gone away for the holidays. And so it was actually left to Jim to check on these mice, and shortly before Christmas, I think, he saw the tumors are growing, maybe nothing's going on. Kind of sad about that. It's not a happy thing to see, but that's what the data was.
We had Thanksgiving and Christmas together for probably, almost 20 years. I remember that Christmas. He was mumbling about, the student taking off and going on holiday.
I had family visiting and everything, and so I did something that was unforgivable, really. If he had done it, I'd have fired him probably, but I skipped a couple of readings for the Christmas holidays.
A few days later, though, Jim remembered, oh yeah, I'm supposed to be checking on this guy's mice because he's still off celebrating Christmas with his family, and came back to find that, in the mice that had received the antibody, the tumors were gone.
It was one of these, truly one of these aha moments when I went, oh, Jesus, this is something big. Because I've been doing tumor immunology enough by then to know this experiment was so cool, you didn't need a statistician. They were all alive or all dead,
These tumors have disappeared. I mean, this is impossible. This is not possible. That's how mind-boggling you have to think of this as, because the human body, with 480 million years of evolution behind it, can't get rid of this stuff. And now the tumors have disappeared.
From then on, for the next decade, essentially he had one goal: to get that made into a drug for cancer patients.
It didn't come out of cancer, but once it was there, it was there. And I'll be damned if I'm going to let that just lay around. This is a way to cure cancer. I knew that was a way.
Well, I'm a kingpin, buzzing round your eye. Well, I'm a kingpin.
In my teen years and in my college days, I was the dork, the one that studied really hard, got good grades, because I was the first in my family to go to college. I had a lot to live up to with that. So I had to make sure that while friends were out partying, their grades were C's, I needed to get A's, and I knew it. Knowing what I know now of what life had in store for me, maybe I would have had a little bit more fun. I probably would have.
We started her on chemotherapy, and that chemotherapy really didn't work for her. So we tried another kind of chemotherapy.
I know that chemotherapy is supposed to kill cancer. It can give you back your life. Chemotherapy made me feel like I had cancer. I felt like death warmed over. I got severe peripheral neuropathy from the chemotherapy. That's numbness and tingling in your hands and feet. It wasn't just hands and feet, though. It went up my leg, and I couldn't hold a pencil to write. Your skin peels off. All of it peels off. So it looks like you have a massive sunburn everywhere. I was itchy everywhere. It was like an itch that you could never stop.
And that chemotherapy didn't work for her. And then I sent her to get high-dose interleukin-2.
My chest started to fill up with fluid. I remember sitting across just like this from my mom in the family room of her house, and I would have to go, Mom, what are we going to do? Dude, that was my breathing.
And the high-dose interleukin-2 didn't work.
Same time, though, I had an MRI done, which I had not had done previously, and they found a brain tumor.
So she needed radiation to her brain.
Do you know when you break your neck and that thing sits on your shoulders and they screw it into your head? That's what they put on you when you have to be bolted to a table when they give you brain radiation. So I had somebody come at me with a drill and screw things into my head while fully awake and conscious.
The life expectancy of someone with metastatic melanoma at the time was about 7 months. So we are way into that at this point.
We published the paper in 1996. Everybody kind of went, wow. And that was kind of it.
We were all very skeptical of the science of cancer immunology.
I wasn't sure that what he had done successfully in a mouse, in a very controlled setting, would ultimately translate to a method that would work in a human being.
You're going to be hearing a good deal more about something called interferon these days.
Skepticism about an immunotherapy drug for cancer stems from the 1980s.
Researchers are now calling a new cancer treatment the most promising in 30 years.
When attempts at using the immune system to fight cancer offered enormous promise.
The drug called interleukin-2.
And then, devastating disappointment.
Because fewer patients than expected seemed to benefit.
For years, there were clinical trials, and none of these worked.
We started to see some bad side effects.
And I can tell you these were not happy meetings to attend. They all started with, we did this experiment in animals in our lab, and it looked great, and then we did a clinical trial, and nobody got better. Years of that had led to a view that immunotherapy would just never work.
20 or 30 years ago, cancer was not a very big thing for drug companies. The drugs didn't work that well, the patients didn't live that long. The much more lucrative drugs were for heart disease or diabetes, chronic diseases that people would take drugs for the rest of their lives.
Jim first goes to BMS, who had held the patents, and Peter Lindsley had worked for them at the time. The guys at BMS had told him, we like CTLA-4, but we think it has other applications. But in fact, they weren't doing anything clinically with it at all.
I was very disappointed in the initial response, but it got more and more frustrating with time. It was just no, place after place after place. The scientists would get happy, and excited, and attorneys would say, no, you can't do this because Bristol Myers Squibb already has filed for a patent on their antibody. And I said, have you read their patent? Their patent is backwards. But still, the company would say, well, BMS has got more lawyers than we do. They're a big company, and so, too bad.
So there were all kinds of things that stood in the way of this happening. And I would say, his experiment in this was to just keep fighting to say, there's something here, there's something here, there's something here.
And that's where I really give him credit. It's like a bloodhound on the scent. He's not going to get taken off the scent until he's found the fox.
model is kind of hard to control because I'm injecting life into myself initially in terms of because the mice, they always have a...
I would always plan vacations, places where he could not get a phone call and he could not be connected to the internet.
Dad is doing a great job of chopping wood. Robert, go over there and stand by Dad. Okay.
It was about 2 years that nobody would listen, and so it was getting depressing. My brother Mike had metastatic prostate cancer. He presented with a lot of joint pain, which is how they found him.
We often had Thanksgiving with Mike. For many years in Berkeley, he'd come out, and so I got to know him quite well. Mike got diagnosed in his 50s with metastatic prostate cancer to the bone. That's how he was first diagnosed, and that pretty much is a death sentence. And that also, during this time, while Jim was trying to march forward this new therapy, he really wanted to be able to give this to his brother.
I need to get it into people. I need to get it into patients.
Jim turns his search towards smaller and smaller biotech companies, looking for anyone willing to take a gamble on his idea. In Boulder, Colorado, he finds Alan Corman, who is creating drugs for autoimmune disease.
My interest was in immunology. I didn't really know much about cancer biology or cancer therapy. I think it was useful not to be biased by the current thinking of how to go after cancer.
The concept of jumping into a problem and being the naive one is really useful. The people that have been in that field very long and can doing the exact same thing, I think you get tunnel vision.
Corman's job will be to take the CTLA-4 antibody that Jim and Max developed for mice and make it into a drug that can work safely in humans. The drug will be called ipilimumab.
I ended up collaborating with someone that I knew from graduate school, a guy named Nils Lomberg.
Nils Lomberg works for Medarex, a small biotech company based in San Francisco.
I got a call from Alan. We started talking about the idea of ipilimumab. Ipilimumab hadn't been created yet, and I convinced him to let me in on this. I was fascinated.
Jim and Alan Corman and Nils Lomberg all get together at the bar in the basement of the Hotel Boulderado, right in downtown Boulder.
Jim's primary concern was that we move quickly. I think he was very skeptical of the whole biotech pharmaceutical industry. He didn't think we had the urgency. I ended up sitting up with him most of the night with a bottle of whiskey. Out of that conversation, we had a handshake agreement. And with that handshake, we started work back in California, back in the lab. 466 days later, we had ipilimumab in a patient. And I think that's a world's record.
Phase 1 tests relatively small number of people generally for safety. Sometimes they get a little hint of whether the drug's working, but generally it's just to make sure that the drug is not going to harm somebody.
We picked a dose that was, a coin toss that was based, somewhat loosely on what we had done in the mouse, but clearly, we didn't know enough about how to figure out its potency.
When we were developing the phase 1 clinical trials, there were many stops and starts. There were many times when it was wondered whether or not the clinical trial could even start Which tumor type should we do it in? Should it be all comers? Should we focus on melanoma patients? How many patients should we enroll in the phase 1 clinical trial? How much of a budget did we have to enroll these patients? What was the starting dose of the drug that we were going to give? Were we taking the starting dose just based on mouse data, which, by the way, is what we did?
When drugs go into phase 1 trials, their first trials in people, 9 out of 10 or more fail. And that's it. You pull the plug at that first stage.
I ran that clinical trial at that time. It was amazing because 3 out of the 12 patients, was a very small clinical trial, 3 out of the 12 patients showed that all of the tumor went away. We never see this in a phase 1 trial. It's so rare that it's sort of like, finding the pot at the end of the rainbow. You feel like, am I really seeing this or am I imagining it, right? It's just unbelievable.
I became aware that CTLA-4 was the focus of an early set of clinical trials that was, through the clinical trial grapevine, producing a little bit of activity in patients with advanced melanoma, and obviously, I wanted to be part of that.
I remember going Christmas shopping in a wheelchair in the mall where we grew up. That was probably the worst Christmas ever. I remember sitting underneath the Christmas tree with Rob, and I was a huddling mess on the floor. And he just held me and told me to work it out. That Christmas was hard because I didn't think I was going to see the next one.
She comes to clinic and I say, I think that we're really at the point where the readily available standard options are not working for you. There is a clinical trial that you could consider participating in.
I remember Dr. Walchuk explaining it to me in very, very simple terms, that instead of an outside agent like chemotherapy, your body then uses its own immune system to fight off cancer. It allows it to turn on the immune system and recognize cancer in a way that it previously couldn't.
Sharon got 4 doses of ipilimumab.
The treatments were not like chemotherapy. Chemotherapy, you go back 3 or 4 days in that week, you live at that oncologist office, it's your home. This was different. I had a shot in my leg, and then I had a 90-minute IV infusion. That was it. Because I wasn't in chemotherapy either, I didn't feel nauseous. I didn't feel the incredible fatigue that came along with it. I noticed a difference in myself just from changing treatments.
Once a drug is proven safe in phase 1 trials, it moves into phase 2 and phase 3. These trials include far more patients studied over much longer time periods. They can cost hundreds of millions of dollars.
We never could have developed ipilimumab as a small company, even a publicly traded company. We never could have raised that much money.
Medarex has to find a partner with deep pockets, but the big pharma companies are wary of the risk. For any hope of success, ipilimumab needs a champion inside the pharmaceutical industry.
When I left the NIH to go to industry, I went to Bayer that was building an oncology program. I really wanted to supervise the development of IPI. When Bayer declined, I left Bayer after 7 years, specifically to bring ipilimumab forward. BMS was not sure they were going to take it at all, so I came at just the right time. And I was ready to advocate for it. I went to BMS. I said, let me walk you through why this is important.
I must admit I was apprehensive. The fact that BMS had another antibody would have been one of the reasons that a lot of companies had given for not working with us. You know, BMS can kill you.
Stocks to watch today. A big one will be Bristol Myers Squibb.
There were other reasons to be apprehensive of Bristol-Myers Squibb.
Bristol-Myers had a pretty good reputation. Then that started to fade. They did a deal for a drug from a small company called Imclone.
The drug sparked an insider trading scandal that led to a 7-year prison sentence.
Pessimism propelled by news that yet another corporate giant may have accounting problems. Today, it's Bristol-Myers Squibb in the government's crosshairs.
So they went through in the early to mid-2000s a very hard period.
Bristol-Myers Squibb, one of the leaders in the industry, forecast disappointing earnings growth for its second quarter.
So what we were asking BMS to do is invest a healthy amount of money into the belief that these drugs were going to work.
It was against the grain. A lot of our external advisors said we were probably investing too much in immunotherapy because nothing had worked before.
My presentation focused not on the response rate, which was very low at the time, but one patient who had received, I think it was a single dose at the time, that now 2 and 3 years out was still in remission.
The very first public meeting, Rachel made it a point for me to come and sit next to her. And I'll never forget, because I was sitting there and there were some people that had very little understanding of immunology that were making comments and, about what they thought this meant or that meant, everything. I was just sitting there going, oh, bullshit. And I was just sort of under my breath, going, oh my God. And obviously getting agitated. And Rachel, kept looking at me, and afterwards, she says, okay, well, I wrote down every time you started snorting and stuff. And so what about this? What's wrong with that? What's wrong with this, from her notes? And so we went over the notes for a while. It was really a lot of fun.
I was part of the group of people who was following, let's call it the conventional path of cancer research. Jim Allison and the immunology crowd was thinking about it differently. And the mainstream was more or less ignoring that.
Innovation is extremely difficult. There was somebody, a regulatory person on my team, who yelled at me, screamed at me. We don't get it. We're wasting our time. And it was not just the members of the team, because back in the day, most of the doctors were not immuno-oncology believers. That's why it was hard to get patients on the study initially. That's why it took so long. There was like a year where that clinical study could not be enrolled. It was very, very slow.
No one wanted to hear about another cancer immunotherapy agent. It was, forget it, we don't want to talk to you. And you can imagine a lot of doors slammed in his face.
You might argue that there is a defense mechanism against new ideas. They are seen as mutations, as cancerous to a body of thought. You don't know whether the new idea is something potent or deadly. And that's why they're often viewed with skepticism, and that's why it takes a really powerful idea combined with someone willing to push it forward to make it happen.
I inherited predisposition for cancer. My mother had melanoma, my grandfather had pancreatic cancer, and my beloved uncle got glioma when he was 58. So I know that cancer is coming for me. I know it's coming for my family. I'm just about the age my uncle was when he got his brain tumor. The stakes are so high. The opportunity is so great that somehow it just didn't matter when people yelled at me or created barriers. Because we're going to make it work. We're going to make it happen.
I'm still in Berkeley, but the action had moved to New York, where the companies are, and a lot of the studies were going on at Sloan Kettering. I thought, well, there's some not so good stuff happening to my baby over there, and I better move again,
Jim decides he needs to be in New York where the major trials are going on. When I asked Jim why he needed to be there, he said, I went there to be a nuisance. He wanted to stay on the scientists, he wanted to stay on the FDA, he wanted to stay on Bristol Myers Squibb.
Anytime a lab moves, it's always a tough decision, especially if you're moving, across the country. People have roots in a given area. I'd been in the Bay Area for, almost a dozen years at that point. So when Jim decided to move from Berkeley to Sloan Kettering, I think it's a testament to how dedicated all of us were to this work and the promise we saw in it, that I would say at least 90% of the people in Jim's lab picked up and moved cross country. It was a tough move in that, you're moving all your stuff, you're moving a lab. Things in the labs are delicate. We actually lost an entire freezer full of sort of critical, unreplaceable reagents. Somewhere in the desert, halfway across, the refrigerated truck that it was plugged into blew a fuse. So there was some pain to go with that move.
Jim, being in New York, was truly a fish out of water. I'm not sure Jim walked more than 3 blocks from his apartment on the Upper East Side. But, he didn't move to New York to take advantage of what New York has to offer. He moved to take advantage of what Sloan Kettering had to offer.
Putting pressure on Jim is Pfizer, the world's largest drug company. Seeing the progress of IPI, Pfizer has launched a competitive cancer drug, drawing from Jeffrey Bluestone's work.
I went to Pfizer, and they got excited about it. We were on track. I was working on them to make the drug, and things looked really good.
Tremelimumab was being developed by Pfizer, and the early signals for both of the medicines were remarkably similar.
Pfizer came in late when they saw reasons to believe. Very classic. We think maybe you have something here. We have one just like it. We're going to beat you.
The race is on, and watching it closely is the FDA, the U.S. government agency that determines whether a drug is safe and effective enough for patients. The FDA is judging the clinical trials by standards developed for evaluating chemotherapy.
The rules said if a patient hadn't had their tumor shrink by 30% within 12 weeks, they failed.
In Pfizer's trial, the tumors don't shrink after 12 weeks. They grow. Failing to meet FDA standards, Pfizer decides to cut its losses.
Pfizer did this giant trial and just everybody was watching. It failed, a big blow. The biggest drug company in the world just abandons the whole field. I mean, everybody, when we started out, one of the main criticisms we had is immunotherapy's never worked, it'll never work, it's a bunch of voodoo, and you shouldn't even try it. Now here's this big failure.
BMS thought there's only one other in the class. It just failed. We don't want to put more money in. In fact, should we be closing the studies you're doing now?
Many of my colleagues said, this will never work. This is a dry well.
Jim struggles to make it clear that ipilimumab is not a chemotherapy drug and doesn't behave like one.
People go back to their community. As soon as they got back, they'd put them on some chemotherapy drugs that had been shown never to work. But now they're responding to such and such, and said, yeah, right. There's another explanation. That's they're just responding late, which is what I showed you in the mice.
You go back to Jim's early experiments in mice with CTLA-4. Jim observed that the T cells were entering into the tumors, and they were expanding in size before the T cells then were able to destroy the tumors, and then they would shrink in size. So Jim had to convince Bristol Myers Squibb to do a much more expensive, much more lengthy trial.
Unlike Jim, who, to his credit, persevered and did whatever it took, I felt I had done exactly what I normally would do, which is to just let the other folks make the drug. Little did I know at the time that they would give up on it so quickly.
Jim was visibly angry. He'd just go into a tirade. I've got to make this happen, because the easiest, you'll never get fired for saying no if you're responsible for spending a lot of money.
The skepticism was widespread, and frankly, Jim experienced it probably every day.
Every time I'd see him, he would just, you could see his blood pressure going up. That was hard on him during those years, and hard on people around him.
That was a trying time. Jim became rather obsessed. The science and the project just consumed him, and I really couldn't be a part of that. With no science background of my own, our son off in college, we had less and less in common. That didn't mean that we didn't admire each other and care greatly for each other, but we just decided to go our separate ways.
I was afraid that the drug was going to go down, and I invested not just myself, but my family. Yeah, it was a tough time. I think any great, big, disruptive change that you're pushing for is going to take some single-mindedness. Whether it has to be sacrificed, I don't know. I suppose maybe it does.
At Memorial Sloan Kettering Cancer Center, doctors are seeing in patients exactly what Jim has predicted.
Patients were coming in and telling us that they felt well. Even though the CAT scans are showing that the tumors are growing, these patients said, I'm feeling really great on this treatment.
Some people, after they came off of the clinical trials of ipilimumab, began to get better with no further therapy. And this was quite striking.
And so at that point, clinicians like Jed Walchuk asked these kind of immovable, implacable forces, Big Pharma and the United States government, the FDA, to change the trial.
The risk BMS was taking at the time was we were committing to years more of clinical study before we'd ever get approval. In an environment where there was tremendous uncertainty about whether this thing was going to work or not.
We were asked to do another 5 years of expensive, long, tedious trials. There were investors that didn't think this was going to work. We hadn't had a long track record of recent success. This takes a toll. This takes a toll.
My brother was in the final throes of metastatic prostate cancer, and the drug was in trials by then. And there were really good results coming back, but it was just not possible. And that was, it was frustrating. But at the time, I just wasn't so sure about whether it would really, at the end of the day, do my brother any good, because he was so sick by then.
remember him calling me from Dallas when, you know, Mike was dying, and he was in his arms at the time. And you can imagine, you know, what that would do to you.
When I was first told I had prostate cancer, I was terrified because it was a week after my brother had died. I'd been working with the prostate group at Sloan Kettering. So I went over to the oncologist's office. The secretary said he's with somebody. I said, I don't care. Beat on his door and said, can I talk with you? And he said, I said, no, now. You know, I need to talk to you right now. My oncologist said, in your case, because of the family history, I think we better take it out. Anyway, they caught it very early.
Sharon came back for her first set of imaging. I got a phone call from the radiologist. Now, usually, you should know, radiologists don't call with good news. They call to make you aware of some awfulness that they have seen on a scan. And this was a very serious radiologist, very well respected. And he called and he said, Jed, I'm reading your patient Sharon's CAT scans. And he says, I have to ask you something. He goes, what happened to her? I'm like, what do you mean what happened? He goes, what did she get? All the cancer went away. And so that was an important moment for many of us.
With enough patients showing improved health, BMS decides to commit to the full 5-year study and accept the enormous financial risk.
Bristol-Myers Squibb made the decision with everybody in the room then to use overall survival as the endpoint and not to use the typical sort of tumor shrinkage immediately endpoint, but look to see how patients did over a long period of time.
I give the team at Bristol-Myers, and Rachel was the lead of that team, credit for listening to us and for being open to the observations that we were really making on the front lines and to working with us to find a way to capture this new biology.
Most companies and most people in those companies at that time would wash their hands and walk away. And so I give great credit to BMS. They went with the science. They believed the scientist, and they believed the data.
Dr. Walchuk, in his ever so calm voice, said, so Sharon, the man that invented this is here today. Would you like to meet him? Yes!
You know, he calls me up, says, come down. He didn't tell me that. You know, I found out why I was there only when I walked into the room.
Dr. Allison walked in, and I pretty much almost lifted him up off the floor, gave him a giant bear hug. I just couldn't talk. I couldn't talk. How in the world are you supposed to adequately thank somebody standing across from you that you are 100% positive that without them, you wouldn't be here? There is no thank you for that.
Oh, meeting her was amazing because until then, you know, it was just numbers. You know, it was a concept. She was the first patient I'd ever met.
And all we could think about at the end of that was, we got to get a picture. We look around the room, and nobody has a camera. The only camera that we could find was one that they took pictures of a melanoma with to send out and get studied. It's not the best quality in the world, but it definitely memorializes one of the best days ever. Every single scan since, I have another MRI coming up here in about 2 weeks, so say a little prayer. No evidence of disease in every one since.
Now she has her family, 2 kids, 2 beautiful kids. I still choke up when I think about that.
2011, 15 years after Jim Allison made his initial discovery, the clinical trials are finally complete.
I remember the moment when we saw the data. I can see the people around me. It is imprinted like a picture because we knew we were changing the world.
Rachel Humphrey came running up the stairs, breathless, to show me the survival curves, one for the control and one for the patients that received IPI. One curve stayed different for as long as we studied. After years, you start to think that maybe you've cured these patients.
I happened to be at a melanoma meeting at Cold Spring Harbor, sitting next to Harold Varmus. I had my iPad on and kept checking it. And he said, what are you doing? And I said, well, the FDA is supposed to announce its decision on IPI today. All of a sudden, I got this email. It was about 11 on the East Coast, which meant it was 8 in California. And there's Alan in the Metarex crowd holding glasses of whiskey. And I looked at Harold, I said, he said, I guess that's good news. I said, I think that's good news, yes. And he said, either that or they just like to drink in the morning.
Allison and others like him have only begun the thing. Each generation will build on the next. We're not nearly at the end of this story. I mean, we're somewhere in the very first few chapters.
This happens fairly regularly. I get emails, but many times a handwritten note. This is from somebody I've never met. Dear Dr. Allison, 2 years ago today, my 63-year young husband, Richard, was diagnosed with metastatic melanoma to the brain and everywhere else. Thanks to you, your fortitude and determination to prove that immunotherapy had promise, Richard is alive and well, healthy and happy, and has been treatment-free for over a year. Anyway, it says, the words thank you cannot begin to cover the gratitude we have for you and your persistence through all this to see that immunotherapy be brought to people worldwide. Our family is eternally grateful for the extra years we've given us with Richard, a wonderful father, husband. Sincerely, it's from his wife. For sure, it's knowing that we've done something like this what makes it all worthwhile. So, I need a drink.
Roll me up and smoke me when I die. And if anyone don't like it, look up in the eye. I didn't come here and I ain't leaving, so don't sit around and cry. Just roll me up and smoke me when I die. Now you won't see no sad and teary-eyed when I get my wings and it's my time to fly. Just call my friends and tell them there's a party, come on by. And just roll me up and smoke me when I die.
When I played with Willie Nelson at the Austin City Limits Festival, I was scared.
Look, I'm in ya.
It was hard to relax, when I'm playing with my buddies, it's like fun. But when you're playing with Willie, I gotta get this, I gotta get this.
congratulations on behalf of the Nobel Assembly.
Well, thank you so much. I'm so honored. You know, of course, it's a scientist's dream.
We think that the discoveries that you have made that has enabled an entirely new principle for cancer therapy, I mean, we think it's absolutely fantastic.
Me out and build a roaring fire. Roll me in the flames for about an hour. Take me out and twist me up and point me toward the sky. And just roll me up and smoke me when I die. Roll me up and smoke me when I die. And if anyone don't like it, look 'em in the eye. I didn't come here and I ain't leaving, so don't sit around and cry. Just roll me up and smoke me when I die. I didn't come here and I ain't leaving, so don't sit around and cry. Just when I die.
Pam Sharma was tenured at MD Anderson. She was a big name in the cancer world in her own right. It was talking professionally for quite a few years. And then eventually there was a romance. Jim said to her one day, "There's no one who can stand either of us. You know, who else is going to talk about T cells all day? You know, we should get married."
¿Qué tal? Emocionante, ¿no? De verdad, una experiencia increíble el haber vivido en toda la plática, en todas las preguntas y en toda esta historia de vida que nos ha permitido y compartido el doctor Jim Allison. Por hoy ha sido pues prácticamente todo y los esperamos mañana, ya al inicio de las sesiones académicas, desde muy temprano, a las 7:30 de la mañana, con simposiums de cáncer renal metastásico, cáncer de mama, tratamiento personalizado en cáncer de mama, carcinoma ureterial metastásico, carcinoma de pulmón y hepatocelular. Todos ellos desde las 7:30, 8:30, nuestros simposios satélites, y los esperamos porque tenemos una agenda sumamente interesante. Simposios con la Sociedad de Cirugía Oncológica de Estados Unidos, la Sociedad Europea de Cirugía Oncológica de Estados Unidos y las académicas con inmunoterapia. También vamos a tener un canal de cáncer infantil con los simposios de SMeO AMOP, Oncología Pediátrica, y toda una línea de investigación y de conocimientos nuevos en oncología pediátrica. Vamos a tener también los simposios de cabeza y cuello, cáncer ginecológico, cáncer colorectal, trabajos libres, sarcomas, genética en cáncer, mujeres en oncología y la presentación de trabajos libres, así como también un segundo bloque de cáncer ginecológico. Nos vemos mañana, los esperamos mañana. Muchas gracias.